Yes, great. Hi, thanks for having me. What I want to, thanks for the lecture. I really appreciate it. And as a return, I actually teach a course in genomic sequencing in the fall that I welcome you both to take, and we can clear up some of the issues that you might have had in terms of that black box, which is not a black box to people who know bioinformatics and sequencing. Just wanted to share with you this image here, which you can see is the supposed, SARS-CoV-2 viruses and the coproteins. These are two independent lines of evidence because they ran a Western gel which detects the protein, as you can see, correct? So this coprotein here, these are actually predicted in the genome itself. So how does that work with your model if we have basically three independent lines of evidence that the genomic sequencing is predicting the spike protein produces a specific type of amino acid sequence that we can visualize with this cryo-EM and also detect on a Western blot, which detects the protein sequence?
I think we would need to see the methods
Sure,
for
absolutely.
evaluating this, which we wouldn't have time to do now, but perhaps we can follow up on.
Yeah, great. Here's the methods. I definitely recommend you look at it. They actually use the sucrose gradient, which you said was missing in extraction of SARS-CoV-2 in this paper. So they ran a sucrose gradient and extracted the virus, and then had several rounds of the plate assays, which shows the CP damage.
Are you saying they extracted the virus directly from a person?
They extracted the virus, they purified the virus using a sucrose gradient.
With no cell culture?
They have used the cell culture, but why does that matter? Cell cultures, again, cell cultures, if you pass through multiple rounds of cell culture, the virus can reproduce. So if you take the extraction of that cell culture and replate it on another plate, the virus then enters the cells, reproduces again and again and again. If by what you say is true, that there's a toxin involved in this, then the toxin will be diluted as you go down with your cell plating cultures, right? So you go through round 1, 2, 3, 4, the toxin will dilute itself, whereas the replicating virus will keep replicating as you add in from the next cell culture. And then, as you can say, another line of evidence is that you extract it with a sucrose gradient, which is what they have done, aligned it with the genomic sequencing, which we can clear up some of the black boxes that you have issues with. And as you can see, the visual evidence matches, right, what is predicted in the genomic sequence.
Okay, let's get some answers, okay?
Go ahead. Thank you.
Yeah, I don't think Tom and I can really evaluate this, but I think the most important question has been answered, that the sample didn't come directly from a clinical sample where the virus was purified out of that.
But it did originally. It did originally come from.
Let's just get the answer and then we can let you respond, okay?
Well, like I said, we really would need to review the methods section in order to know what we're talking about, and we'd be happy to do that and get a response, or at least I would, if you can send me the actual paper.
The paper has been uploaded multiple times now in the comments, and we can email that to you